to be here with you again today to explain and elucidate on our further positive depression results from the XanaCIDD phase 2A trial of Xanamem. We're just going to wait one more second or two for some additional participants, and then we will get started. Okay, I think that looks like it's about it. So the bottom line from today's presentation, and of course the top line, these results were announced on Monday in an ASX release, is that we have clinically and statistically significant benefits of Xanamem treatment on depression. And importantly, this validates the biologic activity of 10 milligram dose and really establishes for the first time, irrefutably in our view, that the cortisol hypothesis, or that inhibiting the stress hormone cortisol in the brain, really matters from a clinical point of view, and that this approach to therapy of multiple diseases, including depression and Alzheimer's, really has traction and really does work. And so to help me explain all of this, our Chief Medical Officer, Dr. Dana Hilt, is going to present the updated results. We've asked Andy Udell, who has recently taken over as a head of commercial development, to join us, also from the east coast of the US, and I will start and finish the presentation. As usual, it would ask that Q&A is put into the chat. We will not address questions as we go through, but we will save those for the end and potentially group them together. We should have plenty of time to answer all questions, but if we don't, we will endeavour to do so by communicating with you afterwards. The recording of this presentation will be available hopefully later today, or at the very latest early tomorrow. As you know, these slides are already released as an ASX release available on our website. Next slide. We will be making forward-looking statements. I'm not going to read all of our disclaimer, but please take note, and that this can be looked at on our website at Actinogen. Next slide. So our drug Xanamem works by inhibiting local tissue production of cortisol in key regions of the brain by this enzyme called 11-beta-HSD1. But what does that actually do? What you're going to see today in these data is that controlling brain cortisol has durable benefits. And, you know, is that plausible? So the easiest way we can explain it, and this is for sure an oversimplification, but it's that stress in the brain becomes chill when you take our molecule over a period of weeks or months. And what do we mean by that? Well, the effects of cortisol as the stress hormone are really quite well understood. You need your cortisol in times of stress to fight or flee, the fight or flight reaction it's called, and it works in two major different ways. The rapid changes don't depend on slow changes in protein synthesis via genomic changes, whereas you do have slow changes in gene expression and protein synthesis that lead to durable on and off effects. And so it's quite plausible, and we have really shown it now with Xanamem, that by lowering cortisol in key regions of the brain, you get both the shorter term and the longer term benefits of improving inflammation, decreasing cell death, improving neural circuitry, changing the configuration of receptors. And this essentially becomes the chill state of the brain. Next slide. And I wanted just to spend one moment on why Xanamem, or some extra evidence that Xanamem is really a unique drug. And that is that the World Health Organization, which administers the procedure for giving new non-proprietary names, has given us a special class all of our own, because nobody else has applied for an INN name of a tissue cortisol synthesis inhibitor, and thus we are recognized by this naming convention as having a unique class. So there are no other known inhibitors of this molecule, this enzyme, being in development for brain diseases, to our knowledge, and we will be able to use this name from 2025 onwards. So this is, we're not disclosing it just yet because it needs to go through a full review period. Next slide. And with that, I'm delighted to hand over to Dr. Hilt to explain the results further.
Thank you, Steve. Michael, maybe the next slide. So to refresh everyone's memory, exactly the trial that was done, the XanaCIDD trial, this was a trial where we enrolled patients that had residual depression and had cognitive deficit. And it was a trial of 10 milligrams of Xanamem versus placebo. And you may remember, if you've been following the development of Xanamem, that the 10 milligram dose previously has shown activity in Alzheimer's disease. And it also shows excellent target occupancy by what's called a PET scan. So we know that the 10 milligram dose gets into the brain, binds the target, and we've had previous studies that have shown that it's active. And so the primary endpoint in this particular trial was cognition. And then very important clinical endpoints were the MADRS scale. And this is the Montgomery-Åsberg Depression Rating Scale. This is a scale that is used to approve and register antidepressant drugs. Another important scale is the PGIS. This is a assessment of the degree of depression from the patient's perspective. The MADRS scale is from the clinician's perspective by interviewing the patient, and the PGIS is from the patient's perspective, plus additional cognitive tests. So the patients then were enrolled into the study. They were treated for 6 weeks, and then the Medicaid study drug was stopped, and then they were followed closely for an additional 4 weeks. So we have 10 weeks of observation in this study. And this is a double-blind study. No one knew the treatment assignment until it was unblinded after all the data was collected and verified. Next, please. To emphasize the point, there are 2 key clinical endpoints in this trial. One is a MADRS scale, which is a 60-point scale. And this is assessed by an experienced clinician who does a structured interview of the patient, assessing different domains of mood to determine the degree of depression the patient had. And this is by skilled, qualified, valid raters. Importantly, there's also the PGIS, as I've mentioned. This is from the patient's perspective. And this is an overall integrative clinical assessment that the patient's making of themselves. How am I doing? And this would involve their mood, their function, basically what's their overall assessment of their condition. This is important because increasingly regulatory authorities are looking to the patient and the patient-reported outcomes as being very important in the drug approval process. So I think it's really important to understand that Xanamem in this trial in patients with moderate significant depression has shown benefit on both of these endpoints. Right? It's shown benefit on the traditional MADRS scale. It's also shown benefit from the patient's perspective on the PGIS scale. Next, please. So this is some technical background, which is important. The way that these analyses that I'm going to talk about were assessed is using the mixed model repeated measures. Now, I know there aren't many statisticians out there. This is the FDA and other regulatory authority approved method of analyzing repeated data over time. So this is done to the highest standard. That's what I want to make a point. The MADRS and the PGIS data were analyzed in that way. All of the analyses I'm going to show are pre-specified. What that means is that this is not the data I'm going to show is not the consequence of looking at 100 different analyses and then picking one was positive. This is all thought beforehand and predicted, and these were tested in a pre-specified manner. And the 3 specified subgroups we're particularly interested in then are patients that were on a background antidepressant, and we enrolled patients that are on either stable antidepressant medication or had been through a significant trial of an antidepressant and weren't presently on an antidepressant medicine. So that was one key analysis I'll show you. And then the baseline severity, patients that are more severely depressed and moderately depressed. We didn't enroll anyone that wasn't not depressed. In other words, everyone had a certain degree of depression ranging from more severe to moderate. And also, the patients had to have a threshold level of a cognitive impairment in order to get into the study. The p-values that I am showing you are two-sided. So this is the most rigorous test. And traditionally, a p-value of 0.05 is considered significant. Now, that's in a fully powered trial. This is a modest-sized trial. This is what I call a learning trial, even though all of the analyses are pre-specified. So if you have the effect sizes that you see, the magnitude of the treatment effect, most phase 3 depression trials are 150 to 200 patients per group. We're treating 80 per group. So when we have these very nice p-values and strong p-values, that shows that there's a pretty significant effect, because our group sizes are much smaller than what one would use in a phase 3 trial. Now, I'll be quoting or stating something called a Cohen's d. A Cohen's d is a fraction of the standard deviation. What on earth does that mean? Well, if you think about the Cohen's d and you think about what we use in the US, the physician's desk reference, which is all approved drugs, the average effect size of all approved drugs is about 0.3. And that's a clinically meaningful effect size. The effect size of 0.5 is a large effect size. There is a reference at the end of this presentation, Hengartner, and he reviews in his paper the last 20-something drugs that were approved for depression. And his observation is the Cohen's d is about 0.28, 0.29. And so to get a drug approved for depression, it has a modest benefit in patients, yet it's clinically useful and the patients need them and use them. So this puts this Cohen's d, I think, into some perspective for you. Next, please. And this is sort of the top line summary of what we found in this trial, that consistently Xanamem treatment does better than placebo on two different important measures, in the MADRS scale and the PGIS. So in all patients that were evaluable, 165, clinically significant benefit was seen at the end of 6 weeks, which actually strengthened through week 10 and became significant. It's p less than 0.05. And then another way to assess this is to look at response rates. And there are two definitions of response rates. One is a 50% decrease in MADRS. That shows that basically the patient's depression was cut in half, and 34% of the Xanamem-treated patients versus 22% of the placebo patients achieved that. And then remission rate, where the MADRS score goes to less than 10 or less, 26% of the Xanamem-treated patients achieved that versus 17% of the placebo patients. So that's a very nice response rate, and that really shows that it can benefit patients. And the PGIS curve separated earlier and continued to favor Xanamem over the entire treatment period in all of the subgroups that we analyzed. And we find, and this is something that's consistent throughout this study, that the strength of the signal continues to increase over time. And at 10 weeks, it's actually maximal. This suggests, and Dr. Gourlay talked about the fast effects and then the slower effects. We think that the effects of Xanamem are going to be magnified over time, and we see a gradual improvement over time. And so one of the things we're thinking about in future trials, of course, is to think about dosing the patients for longer, because this effect continues to get better. Next, please. So this is looking at who we enrolled into the study. So the mean age of so I'm showing both groups, the Xanamem-treated group and the placebo-treated group. And so what's the age of the patients in each group? Well matched, 49%. Female, well matched, over 60, and that's not unusual. In fact, that's typical for depression studies. Now, we used a scale called the Hamilton Depression Scale to assess whether the patient had residual depression. So the patients had to have some degree of depression to get into the study. And that was well matched as well. We used a cutoff of 17. The mean of both groups was 21. And then the MADRS mean was 25, 24, 26. So these are patients not with completely treatment-resistant depression, but residual depression. And the majority of them were on background antidepressants, as shown in the next row, 77 and 86%. So this means even though they are fully treated, in quotations, with an antidepressant, they still have significant depression. They still have MADRS scores of approximately 25, which is moderate level depression. So that shows you that the standard treatments out there are not effective, certainly in all patients. We also required a cognition deficit in the patients, and we used a screening test, a quick screening test called the box filler, and that was about the same in both groups. One thing that was divergent, and this may have impacted the analysis of cognition in the study, was the attentional composite, or another measure of cognition at baseline. And you can see in the patients who received Xanamem, it was a plus 0.11, and in the placebo group, it was a minus 0.1. Now, to put this in perspective, remember that Cohen's d that I mentioned before. The difference between plus 0.11 and minus 0.1 is about 0.2. So that's actually an effect size that is equivalent to some of the drugs that have been approved for Alzheimer's disease. So these two patient groups were slightly different, and this may have impacted our ability to assess cognition. Next, please. So if we look first, the most important, I think, clinical endpoint from the clinician's point of view, the clinician's assessment of the depression over time, what I'm showing here on the y-axis going down is the MADRS score. And so losing points on the MADRS scale is good. So down is better on this graph. And on the x-axis, from left to right, we see baseline, week 2, 4, 6, and 10. Now, the solid purple line, excuse me, represents the patients in all these graphs. The solid purple line will be representing the patients who received Xanamem. And the gray line will represent the patients who received placebo. And you can see there's a gradual separation. This is a modest-sized trial, so there's some variability. Gradual separation, some separation at week 2, week 6, more separation, and week 10, very significant separation. And you can see at week 10, the p-value is significant, less than 0.05, and the Cohen's d is 0.4. And remember what I said about this paper that's in the references of Hengartner. The last 20 drugs that have been approved for depression, the mean Cohen's d is 0.28, 0.29. So that Cohen's d, if that's a real Cohen's d, and we can repeat that, that's very significant, I think, as far as a treatment effect size for this patient population. Next, please. And we look at the other important clinical endpoint, which is the patient's perspective. And you can see here that this separates at week 2, and this continues to improve over time. And out at week 6 and week 10, we have Cohen's d of 0.23 and 0.29, p-values 0.19, 0.12. So there's a clear effect here as well. And this supports, each of these support the other. So you have the clinician's assessment of depression, and you have the patient's perspective of how they are doing. Next. And another way that is used in depression trials is to look at what are called responder rates. So there are two definitions of response in a depression trial. If a patient has a 50% reduction in their MADRS score, you say they are a very robust responder. So that's the left graph. And what you can see is gradual increase in the divergence between the treated population and the placebo population in the percent with a percent of the populations with a greater than 50% reduction in MADRS. And so that's a very nice result there, and shows that there was a significant difference there between those two groups. Now, another definition of response is remission. So if a patient goes with their MADRS score goes down to 10 points or less, they're thought to be in quote unquote remission. So the residual depression symptoms that they may have some, but they're relatively minor or modest in scale. Now remember, the median or the mean of the population going in was about 25. So that was a significant decrease. And what you see here is, again, a big difference in the remission rate between the Xanamem-treated patients and the placebo-treated patients. Two additional measures to show that the drug is having a beneficial effect. Next, please. Now, this is a complicated slide, so we'll spend a few minutes on this. And so what I want you to get from this slide is the big picture. So what I'm showing here, if for first column, are the different endpoints. One is the MADRS, and so the Cohen's d of greater than 0.3. Now remember, I said 0.3 is a moderate treatment. So what we're saying here is that if the number in the column is green, and those represent the weeks, that shows you the drug is better than placebo. The next row across is the p-value, significant or not. The next one down is the PGIS, the patient's perspective. And this is also the Cohen's d greater than 0.3, a nice solid treatment effect. And the next one down is the p-value of the PGIS. So as you look across here, if you see a lot of green, which unless you're colorblind, you should be seeing, means that the drug does better than placebo and it's significant at those different time points. So for example, in the first row, all at 10 weeks, their significance, 2 and 6, 10, 2, 6, 10, so forth and so on. And so you can see that at many different time points on both the MADRS and the PGIS, you have a clinically significant difference. The Cohen's d is greater or equal to 0.3, and the p-value is less than or equal to 0.05 at those various different subsets of patients in the trial and at different time points in the trial. And so as you look at this as a gestalt, you're seeing a lot of green numbers, which means the drug is doing better than the placebo on all of these endpoints at all of those time points. Okay, next, please. And drilling down even further into some of these subgroups, in the left panel, I'm showing you the mean of MADRS in this entire patient population was 26, okay? So if you look at the patients that had less severe depression, still depressed, okay, but they have 26 or a little less. What you can see on the left is that there's a very robust treatment difference there, with the Xanamem patients doing better than the placebo, separating at week 2 and continuing to get larger, with a Cohen's d at week 10 of 0.87. I mean, there are very few drugs that have Cohen's d of 0.87. Antibiotics do, things like that. But depression drugs, that's unusually robust. And now, if you look at the patients who have the higher MADRS, what you see is eventually they separate week 10, but it takes them longer. So when one sees data like this, it's tempting to hypothesize then that maybe if the patients are less severely depressed, they respond more easily earlier on. But if the patients are in a really deep hole and they have pretty significant depression, it may take longer for the drug to produce a benefit for these patients. But you see at the end there, at the right, the Cohen's d is 0.61. That's a very nice difference. Next, please. And these are the corresponding traces then for the PGIS. And you see here in both groups, the less severe and then the more severe, the PGIS separates as early as week 2 and continues to stay separate, with the Xanamem treated patients doing better than the placebo-treated patients. Next, please. Then finally, I'll turn to safety. As you may know, this drug has been in a fair number of patients. More than 350, close to 400 patients have been treated. And what we saw in this trial is that if you look at the treatment emergent adverse events, the table at the bottom of this table, and we take any treatment emergent adverse events that has an incidence greater than 5%, and those are headache, fatigue, nasopharyngitis, upper respiratory type symptom, and a frank upper respiratory tract infection, you can see the frequency there in the Xanamem-treated patients of 82 patients and the placebo-treated patients, 83 patients. And those results are very similar in all of these categories. In fact, there were more patients in the placebo group with headache than in the Xanamem-treated group. Very importantly, the third line down is serious adverse events. These are regulatory defined adverse events of special importance. And you can see that Xanamem had 0, the placebo treated patients had 1. And as an aside, during the entire development program of Xanamem to date, knock on wood, we've had no serious adverse events that have been associated with drug. So I think it's quite fair to say now, as we gather this growing body of evidence and data, that the drug is very well tolerated. There are no serious adverse events associated with the drug. And so this suggests a drug with this type of safety profile could be used even as initial therapy for patients with depression, and probably could be easily added on to patients who are presently taking an antidepressant but aren't getting an optimal response. Well, that's an awful lot of the patients with MDD and with depression. And so I think this is a very nice aspect of this drug that will help clinicians and patients use this drug. Next, please. And next. So a few concluding remarks. We're very pleased with this data. Who wouldn't be, frankly? There are clinically and statistically significant effects on depression. And these are from 2 viewpoints. You have the patient's viewpoint on MADRS, and the clinician's viewpoint on MADRS, and the patient's perspective on the PGIS. And they're also supported by these improvements in higher responder rates by 2 separate definitions in the Xanamem-treated group versus a placebo-treated group. The drug was safe and well tolerated. We did not see any significant safety issues in this study. And in many of these traces, you will see that in graphs and analyses, you noticed that the Xanamem-treated group continued to get better from 6 to 4 weeks after the medication was stopped. And when we think about the potential mechanisms of Xanamem and how the blocking of cortisol in the brain is very different than just manipulating neurotransmitter levels, which all the other antidepressants do, this may be suggesting that we could be having more durable, prolonged effects. And this will be important to look at, I think, in subsequent studies. This could be re-engineering the brain or repairing the brain versus just adding more fuel to the tank by altering neurotransmitter levels. Next, please. I've already mentioned this, but there may be this evidence or suggestion then of a durable effect, and it really validates this 10 milligram dose, and we have quite a bit of experience now at this dose, and it's very well tolerated. And this extended effect suggests there may be some disease-modifying benefits. This is what we're looking at in the Alzheimer's study, which is ongoing now, which is going to be looking at patients over 36 weeks. But I think it's very important to understand that looking at a study in patients with MDD is very different than Alzheimer's disease. This trial was 6 to 10 weeks, 6 weeks of treatment, total of 10 weeks. Most depression trials are from 6 to 10 weeks of treatment. All right. Alzheimer's disease trial would be treating the patients for 36 weeks. Depression trials looking for an acute benefit in depression and mood. The Alzheimer's trial will be looking for a longer slowing in the rate of progression of the disease. So they're really apples and oranges. And this shows you the drug may be manifest its clinical benefits in different ways in different patient populations. Next, please. And just a quick comment then about the scientific rationale. We haven't had time here to go into this. There is a reference in the references by Professor Seikel, which really comprehensively reviews this, but there is a vast literature on the potential deleterious effects of cortisol in depression, the fact that cortisol can be neurotoxic to neurons, that there's elevation of spinal fluid, CSF, and plasma cortisol that is associated with worse depression, more difficult to treat depression, patients who relapse with very significant depression. And there was initial positive effects of a pan-cortisol receptor antagonist, mifepristone, which is completely not well tolerated long-term, can't be given as a therapy, but that had antidepressant effects. So there's a wealth of background to suggest that, yes, one should expect if one could block brain cortisol production, one might well predict that it could have antidepressant effects. And lo and behold, that's what we did, and that's what we showed. And so I want to turn that, on that note, over to Andy, maybe talk a little bit about patients with depression, how they're treated, and how this might fit into that picture.
Thanks, Dana. So focusing on the right half of the slide, according to the National Institute of Mental Health, looking at a 1-year period, 21 million patients have had one or more MDD episodes in a past year, 2/3 of these with an episode with severe impairment, and 61% of all adults with MDD episodes receive treatment. And they've generated greater than 365 million prescriptions per year. So as you could do that math, you could see that they are receiving, as Dana said earlier, multiple medications, and this is not uncommon in depression. So in summary, we have a new mechanism of action with some scientific and clinical evidence in a very unsatisfied market. So now we have many antidepressants on the market for MDD. So you may wonder if we have room for one more. So let's look at the next slide, please. So let's take a look at the latest launch of an antidepressant that's indicated for MDD. The product's called Auvelity. It's promoted by Axsome Therapeutics, and it's a combination of dextromethorphan and bupropion, which are two generic drugs that are out there in the market. It showed a similar type benefit in MADRS versus placebo that you see with other antidepressants that are approved. And once again, it's indicated for MDD in adults. What they're really focusing on in promotion, and what's unique about this product, is they tout that it started working for some people as early as the first week. And the other thing that they really focus on is it works differently. So you start to wonder, how is their launch gone? And to be clear that this product is highly managed by payers. And Axsome is not exactly, big pharma out there pounding the pavement in the States here. So go to the next slide, please. So what we see here is Axsome's really had a successful launch. In the first full year on the market, they generated over 130 million net sales. The most recent quarter has been 65 million in net sales. So they're on pace to have a very solid year, well over 200, probably closer to 300 potentially $million. And they still estimate a peak sales in the US of between 1 and 3 billion. So I think, we would look at that as certainly a successful launch. And I think looking at the differentiation and of that Xanamem presents, looking at the scientific, the new mechanism of action, and the potential when we speak to KOLs that we've shown some of this data to is pretty exciting and gets them as well as us excited. With that, I'll turn it back over to Steve to finish up.
Thanks very much, Andy, and to Dana for those great explanations, particularly taking us through the detailed data of those excellent results. So what are our next steps? Obviously, there's a lot of exciting stuff to do. This is a really big deal to have, as you said, Andy, a drug with a safe and novel mechanism. It differentiates itself. It's in a unique class. It has potential not just in depression, but in Alzheimer's and other neurodegenerative diseases as well, potentially other neuropsychiatric diseases. So, however, we can't do everything all at once. So our immediate focus is to enroll the Xanamem phase 2b trial in Alzheimer's as quickly as we can. That trial is up and running in Australia at 15 clinical sites. I visited one of those sites that's very enthusiastic about the cortisol mechanism and the recent depression results, indicating a higher probability of this drug working in Alzheimer's as well. We are opening US sites next month, and those will be up and running in short order. We're implementing product development plans both for Alzheimer's disease and depression, with very much a focus on designing a program that can speed our path to marketing approvals. And obviously, we'll be having partnership discussions along those lines as well, because additional resources from partners would certainly help. We need to finalize details of our data analysis from this current study, in particular details of the depression responder patient population, although I think we've largely got a handle on that, in that the drug is having fairly broad effects. And then we'll consult broadly. We've already talked to multiple local and global depression thought leaders. We'll be talking to more consulting pharmaceutical partners, several of whom we've had discussions with already, design the phase 2 protocol and discuss that with the FDA, and conduct the meeting later this year or early next. Andy is working with us to define the commercial product definition for our drug for MDD, because Xanamem is now going from early clinical development, where it's really about does the drug work, to late clinical development, which is all about developing the features and confirmation of Xanamem as a product for various indications such as MDD and Alzheimer's. Manufacturing will continue with the manufacturing of additional tablets to supply the pivotal studies, which will be phase 2b, 3 studies in MDD, but also, of course, in Alzheimer's disease. And then just to finish off, this slide's been shown before. To summarize where we are next year, we should have interim data from our Alzheimer's phase 2b in the middle of, if you can go back, please, Michael. Interim data in the middle of the year, with final results about a year later. With the positive interim data, we would hope to start a phase 3 in parallel with that. For depression, we'll go into a period of consultation, as I mentioned, over the coming few months, with a plan to start a pivotal phase 2b/3 study in the first half of the year or towards the middle of the year. A number of other ancillary things, including manufacturing, clinical pharmacology studies, need to be done. And of course, we're working hard to make sure all of our data is published in peer-reviewed, high-quality publications, and that still is ongoing. So I'm going to finish up there. We have a few questions in the Q&A. We just go to the next slide. Oh, sorry, I forgot to mention the links in this reference page are live, and I would encourage those who are interested in the technical aspects of particularly 11-beta-HSD1 inhibition. Jonathan Seckel, the endocrinologist from Edinburgh, has written a brilliant paper, very detailed on all things concerning 11-beta-HSD1, and it's a very, very good paper. So on to the Q&A. So we have a few questions, and I'll just try and take them all in order. David asked, what is the revised estimated market opportunity now for depression, and I think, Andy, you may well have answered that already. Our analyst at Edison estimated a peak sales potentially for our drug of about $2 billion a year. You gave a range for Auvelity of 1 to 3. Any other thoughts to add to that question?
No, I think that's, you could see how that drug has done with who's promoting it and how unique it is. And I think we have a very large opportunity in such a large unsatisfied market. So that's the best I would give you right now as well.
All right, thanks, Andy. Peter asked, will you be doing more follow-up? And Peter, I assume you mean, will we be able to follow up these patients in the XanaCIDD trial for longer? Unfortunately, that's not really practical. The patients were originally consented for a specific trial. You can sometimes go back and reconsent patients. However, I think that the prudent plan for us is to move forward with a new clinical study with a longer period of treatment and additional follow-up in the future. Dana, you're nodding. Anything else to add?
No, I think this is what the outside experts that we've consulted so far have said. They've all said, that's very interesting that your effect seems to be getting larger and larger. It would be very interesting to treat patients for longer and then to follow up and see whether those benefits persist after you stop the drug. Are you having some, dare I say, even disease modifying effects in patients? So these are big questions that we need to address in future trials.
Yep. So I have several questions from Alan. Alan sent a couple of questions ahead. Let me just, Alan, I'm going to answer those ones and then ask the question you just asked. So first question Alan asked was, in the next trial, would it be sensible to do cognitive impairment and MDD as joint primary endpoints? So Dana, I don't know, would you like to answer that one?
I think one of the sort of lessons you learn in drug development is you go where your signal is. And, I think what this data is telling us loud and clear is that we have a MADRS signal, we have a PGIS signal, we have two separate views on depression. And as Andy has said, there is room in the market for a new antidepressant drug with a very novel mechanism of action. So I think we'll be focusing mostly on depression. I don't know, Steve, if you want to make any comments on that. But...
Yeah, no, I think that's exactly right. Alan asked also, do we have the team to pivot from cognitive impairment to MDD? And I'll take that question. Alan, we have a very deep and experienced team in drug development, including the clinical operations required to run complex neurologic trials. So the Alzheimer's trial requires careful training and measurement systems to make sure quite complicated endpoints, such as the CDR sum of boxes and other things, are adequately measured with low noise, etc. MDD is very much the same. In fact, with less of a focus on cognitive impairment in MDD trials, the trials are actually going to be significantly simpler, as these endpoints like MADRS and PGIS are relatively well accepted, and people have been trained on them fairly widely at clinical centers. So it's actually simpler for us, indeed, to focus on MDD, as Dana just explained. And your last question was, could you please remind us who is Andy Udell? And Andy, I'm going to let you remind Alan who you are.
Sure. So that would be helpful, probably. I've been in the industry well over 20 years. Most of my career has been working for biotech companies, helping them move from clinical stage companies to market-ready commercial companies and fully integrated companies. The first company I worked for happened to be a small company called Clinical Data Inc. And I was the only commercial person working on an antidepressant, vilazodone. And we took that from phase 3. I joined the company in phase 3 all the way to launch ready. And after approval of the drug, we actually sold that company to Forest Labs. And that drug's on the market to today. Vibrate is the brand name. And most recently, I worked for a small company, a Swedish company. I was the president of the company, helped them move from 10 people in Stockholm and me to a fully integrated company that launched a rare disease product, got it approved and launched it last year to over 100 million in net sales. And the company recently sold to a Japanese company called Asahi Kasei.
Yeah, thanks, Andy. And so, Alan, you can see that Andy's got a great deal of experience that's perfectly relevant to helping us move Xanamem forward from the early stage, clinical stage that I talked about into the maturing later stage and commercialization stages. So thanks, Andy, for that. Dev asked, can you shed any light on the responsive expression, response of experts and potential pharmaceutical partners that you've consulted? Dana, do you want to just maybe give a little bit of color on the experts?
Sure. So we've talked with 3 or 4 depression experts. I'm not going to name names, but including one that is done literally as an investigator and then running a CRO, probably in the triple digits. All of them think that, yeah, there is definitely an antidepressant effect here. They're all intrigued by the persistent benefits and the fact that this may have more prolonged benefit. One leading academic expert in depression said, oh, gee, this seems to be the first new mechanism for depression in the last 20 or 30 years. This is a big deal. So far we've had a very positive reception, but we do need to assemble a bona fide clinical advisory board and have the way I'd like to do it is talk with them individually and then bring them together and really have a discussion about what's the next steps, what are the best development options we have, the best treatment population, the best designs for studies. And these are things that we'll be doing, and we'll be doing this in parallel with soliciting FDA and other regulatory advice and input on next steps and what they would expect us to do to get the drug to the market. So that's from the expert side. What was the other?
I'll do the partnering side.
Okay.
Yeah, look, it's a little bit, well, first of all, partnerships are a very private thing until they're done. And so, they take, several months, usually at least to, 5 or 6 months to do any kind of a partnership. What we can say at this stage is that people are very intrigued and interested in the results, and those meetings are ongoing. Justin asked a good question. What's your hunch if the trial was actually longer? Would you have actually hit your cognitive endpoint? Do you think, Dana?
I don't know. This is my opinion. You'll remember that I said there was that difference at baseline between the two groups. I think when we look at the placebo response in cognition in this trial, it was unexpectedly large, much larger than we had seen in other studies. And so I worry that really impacted our ability to detect a cognition signal in this study, and really clouded the issue. I don't know, Steve, if you have any thoughts or comments.
Yeah, I think it's very hard to say. I think Dana's original comments are the most important. We have a very strong signal on depression itself, with different measurement systems. So at least in patients with depression, I don't think this drug is primarily a cognitive enhancer. I think this drug is modifying underlying biologic processes consistent with the known pharmacology and biology of cortisol. So I think that's where we need to focus. There's a few questions from various people about partnering, and I think I maybe partly answered some of those already while we were talking. Obviously, when would you expect to announce partnerships? We have a duty of continuous disclosure to the public markets, and as soon as we know, everybody else will know too. Is there a timeline on that? As I said, it commonly takes many months to do a partnership deal. It's often a bit of a dance as you develop a champion inside the other organization, go into multiple layers of different levels of discussion. So it's certainly not something that can be done overnight. Philip asked the question about, do you need additional funding to do your future trials? So fortunately, and thanks to those who've participated recently, we raised money in the last financial year of a bit over $19 million that's given us a runway through towards the end of 2025 to cover our ongoing. We've just about closed out the XanaCIDD depression trial. We're now ramping up a little bit with US sites, the Xanamem trial in Alzheimer's disease, and we are covered with the operating funds through the late part of 2025 and past the interim analysis. So, however, obviously, for the phase 2b study in depression, that's something we very much seek partnership contributions and for as co-development or some other form of cooperation. So that remains to be seen. David had a question that's a good one. He said, I've experienced side effects when stopping antidepressant therapy cold turkey. Have any observations been made of the effects of stopping Xanamem? Dana?
No, and we can look in the 6 to 10 week time window, and we really haven't seen any significant adverse events. And this is one of the, I think, signatures of this drug, knock on wood, to date, is been exceptionally well tolerated and has very few side effects at the doses of 10 milligram or even 5 milligram. 5 and 10 milligram been very well tolerated, and we haven't seen any of those manifestations.
Yeah, thanks, Dana. My analysis of various trials over the years has been exactly the same. I think there's quite good evidence that there are no adverse problems with stopping Xanamem in any of the trials we've done. So there's a few questions about the cognitive story, and I think this is probably good, Dana, if you can sort of like reiterate again sort of why we're so excited about depression as opposed to cognition in depression. So first asked, was the cognitive outcome just bad luck? Can you speak to that? The second relevant question was, are you moving to depression because it's an easier path? And maybe that you speak to those two questions.
I mean, as I said before, one of the axioms in drug development is when you have a signal, you follow it. And so the drug has shown a very strong antidepressant effect from two different perspectives and measures, so we're going to follow that. As far as cognition, I don't want to get too technical, but in our 2 normal and older normal volunteer studies where we showed a procognitive or benefit of cognition effect, we had a placebo effect of 0.15 units, okay? And what we showed in this trial, what we had in this trial to deal with, was a placebo effect of 0.4 units, more than twice as large. And so I think, this very robust placebo response drowned out our ability or swamped our ability to see a cognition effect. We won't know. We don't know now. But as Steve has mentioned, we're planning on following the depression signal since that was evident by two different measures. I think that is basically what we can say about it at this point.
Thanks, Dana. And then Alan asked another question about confounding by background antidepressant therapy. Were the results confounded by being some being on combo therapy and some on monotherapy?
But what we did have a stronger effect, Alan, in patients who weren't on background therapy. Now that's a minority of the patient. There were 31 patients that were not on background therapy, and then 134 who were on background therapy. Now, the ones that weren't on background therapy previously had been in at least a 6-week trial of a bona fide antidepressant. So they're not treatment naive. But it stands to reason that maybe if patients weren't on background therapy, you might be more easily able to show a signal. But the drugs seem to work in both patient populations, and there is need in both patient populations. You know, one of the you might ask, well, why are patients not taking antidepressants if they have chronic depressive condition? Many patients don't tolerate these. They don't like the way they make them feel. They have a variety of side effects that are not well tolerated by some patients, and so they just won't or cannot take them. So there's need in both sectors, both in patients who are on antidepressants, supplemental medications to give a better treatment benefit to the patient, and patients who really don't tolerate antidepressants that well.
Thanks, Dana. That's good. All right. Well, we're just about at the top of the hour, and I sort of last question from my our investor and colleague G, who is a big believer in the company, noted that it's my 65th birthday today, which is true. And was I happy with the results as my birthday present? And the simple answer to that question is very much so.
Well, happy birthday.
Thank you. When I took the job as CEO 3.5 years ago, I did so after 3 months of diligence and a deep dive into the data, and formed my own conclusions that this drug was a safe, good-looking small molecule pharmaceutical with activity in the CNS that was going to work. And I do feel that these results we've now just talked about are the proof of that, and there are great things ahead for this molecule and for the company. And so with that, I think we will remind everybody that the recording of this will be available hopefully later today or tomorrow at the very latest. And we thank everybody for your participation.